All right, I'll try this again. I do not think that my last one was posted. I've had some computer problems, so let me go through this one more time. There are two things that I want to focus on. One is this action potential. How do we actually generate it? What does all this mean with this trace? And then I want to talk about what are called support cells or glial cells. So here's how this essentially works. That patch clamp is going to read to us the fact that the inside of the cell, on the inside of the membrane, sits at -70 related to the outside of the membrane. Now what we have to do is we have to somehow get to that -55. That's our threshold. That's literally like pulling a trigger on a gun. You can't bring the bullet back. So once you've hit here, that's what's going to happen. So what's going to essentially occur is this is a chemical gated channel. So when a neurotransmitter, that we'll talk more about later, when a neurotransmitter ends up binding with the outside of the membrane, it opens sodium channels. Sodium is much more prevalent on the outside than it is on the inside. So it's going to move in, it will diffuse. As those sodium ions go in, it carries a positive charge, makes it less and less and less negative until we hit -55. This is when we have what are called voltage gated channels open. So initially it was chemical gated, now it's voltage gated. And the sodium gates pop open, all the voltage gated ones. Now, all of a sudden, sodium rushes in. It just takes off, going into the going across the membrane into the cell, and that's why we see this big spike. Once we hit 35, plus 35, what's going to happen is this: those voltage-gated sodium channels are told to shut, and they do, so we don't have a flow of sodium anymore. But we open potassium channels. Potassium is positive, and potassium flows out, and it takes all of that positive charge with it. As the positive charges leave, the membrane potential drops. And then we have this little dip down here, because what's going to happen is this: the potassium is moving out very, very quickly, and it actually goes shooting past that resting membrane potential, and then we have what are called sodium-potassium pumps, and those pumps fire up, and what they do is they send let me make sure I get this right, the sodium back out and the potassium back in, and so they maintain that -70, and then it waits until there's another signal that gets it up to the -55. At that point, you pull the trigger again, and you're off to the action potential races. Now, a big question might be floating around, well, how do you get there? How do you hit -55? How do they talk to each other? Well, essentially, here's how. What we've been talking about are what are called action potentials, but there are also what are called graded potentials, or some of the time they're referred to as local potentials. So picture all of this input from all of these different neurons. It's not just one neuron talking to another. It might be 500 talking to one. And what's going to happen is they are going to generate what are called EPSPs or IPSPs. And here's what this means. This acronym, the first one is excitatory, which means getting closer to the threshold, postsynaptic potentials, which we really haven't talked about synapses yet, but we will. And IPSP is inhibitory postsynaptic potentials, which means those are driving this further away from hitting this threshold. So what's going on with your neurons right now is this. Their membrane potential is bouncing back and forth. It's getting higher, getting lower, getting higher, getting lower. But what counts is if it gets to here. So you might wonder, well, how does a neuron tell another neuron to get closer to being excited, the EPSP, or further away from getting excited, the IPSP? Essentially, this comes back to ions. If I am going to try and excite you and get you closer to -55, I'm going to generate an EPSP. What I'm going to do is tell your chemically gated sodium channels to open. And so sodium rushes in, you get closer to -55. If I'm trying to inhibit you, what I'm going to do is tell you to open potassium channels. Potassium moves out and this line traces down. So you've got these two things going on all the time. And it's what we call summation. These neurons are summing. They're taking input from excitatory, inhibitory, and then they have to figure out, do I hit 55 or -55 or not? If they don't, they just bounce back and forth down here. If they do, they generate an action potential. Now, to play off of these EPSPs and IPSPs, I said I'd explain what a synapse is. And when I have one neuron, and this is kind of reminiscent of the neuromuscular junction, where we have a neuron dealing with a myofiber. Let's say I have a neuron. There's the end of the axon, that axon terminal or bulb. And let's say this is a dendrite down here. They don't touch. There's a gap. This tiny gap right here is known as a synapse. Our brains, these incredible machines that we have in our cranium, have been estimated to have well over 1 trillion synapses. So there's a lot going on up there right now. Even when you're sleeping, if you've ever had a dream, you know what synapses are all about. They talk and they talk and they talk, and they generate all sorts of images and sounds. But they have to somehow get the information across that synapse. So we have a neuron that is going to have little vesicles with things like serotonin, dopamine, what we call neurotransmitters. When they are secreted, when they're exocytosed and they're picked up by the chemical gated sodium channels, or potassium for all that, or for all intents and purposes, what it's going to do is tell this neuron, this postsynaptic, this EPSP or IPSP, that hey, you have to start moving ions. And so you can excite it, you can inhibit it, but keep in mind, you've got another one, or two, or 2,000, or 200,000 right next to it. So if this is an EPSP, it might cancel out the IPSP, which all of a sudden, this is where thinking comes from, this is where critical thinking comes from, this is where memory comes from. So we have to do what's called summation. These neurons will take into consideration all the EPSPs, all the IPSPs, and figure out, do I hit negative 55? That's the gold standard. Sorry. So this is what summation is about. This is about how we generate action potentials. This is what EPSPs are, IPSPs are, and it all goes back to ions. So in the very beginning of the semester, you start talking about ions, and you didn't think they were important. Well, yeah, they are. So this is why. Months later, we're still talking about them, and this is how we generate a nerve impulse. When we talked about, or I mentioned action potentials going on in the muscles, and we've got this action potential that shoots down a T-tubule, it's this. I believe that the numbers are slightly different, but not much. But it's all about chemical gated, all about electrically gated or voltage gated channels. So with that being said, what I'd like to get into on the next video happens to be the support cells. You know, we put neurons up there as a gold standard, and they're incredibly important, but they're not the only cells that play a big role in the central and peripheral nervous system. Central being the spine and the brain, peripheral being all of those that shoot off of there. So let's deal with those and see if we can finish up the nervous system.